It's a great pleasure for me now to welcome to the podium my friend and colleague, Professor Vicki Whitehall. Vicki is one of the senior group leaders at the QIMR Berghofer, the Medical Research Institute just down the road. Vicki comes from a background in science. She runs and is the group leader of the Conjoint Gastroenterology Lab at QIMR Berghofer. Vicki has worked hand in glove with gastroenterologists and surgeons and oncologists for decades. And is really the powerhouse behind a lot of the work that they do. And I think today, Vicki, you're going to share with us from the scientist's point of view how a marriage, if you like, between clinic and science works in real life. So, Vicki, over to you. There you go.
Thanks, David. Yeah, so that's true. I've trained as a scientist and I've worked with clinicians my whole career, and I've been very fortunate to be supported by really outstanding clinicians and outstanding researchers. And I want to share with you a bit of that background today. I'm going to tell you a little about what it's like to have a career in science, and also share with you some of my amazing mentors and how I've worked with them to do translational research. So science is actually really fun. I've always thought it was fun. It's exciting and it's very rewarding. There's lots of variety. There's great opportunities to travel, which is particularly good when you're young, before you have a family. And it's quite flexible. But there are some challenges, as with any career. It's quite difficult to obtain funding, both for your own salary and for the research you want to do in the lab. So that can be really quite challenging and can mean that scientists need to change jobs and fields and be ready to pivot from what they thought they were going to do. It can be really hard when an experiment fails or you don't get the result you anticipate, but you just go with that. It also can be difficult for women and to balance when to time having their children. So that's something that you need to consider when planning your career. So as a scientist, you start off at university, you do your Bachelor of Science. If you want to go into research, you do honours, which is 1 year research, then a PhD, which is 3 or 4 years research. You then do postdoctoral training, and in amongst all these points, you might work as a research assistant or get extra experience in labs. So this could be quite a protracted process. Generally, you would go overseas to do postdoctoral training for a couple of years, 5 years or so. And then you would hopefully come back to Australia and bring your knowledge and learning and become a junior group leader and start your own group in an institute in Australia. So by the time you've done all this, you are quite specialised in your chosen topic. You have, and I put this here to give an understanding of the expectations on scientists and also what they can contribute. So by this stage, you really have really highly developed expertise in what you have chosen to do, and in the process of finding data and integrating data. So I think that's a real basic skill of scientists that you need to, when you have a new question, to go out and find where the information is and put it together to work out what needs to be done and what's novel and what's not. And you need to do this to formulate interesting and important research questions and make sure that your question is going to have a defined outcome. You need to write research grants to make sure you have money to do the work. You need to publish your findings so that it's meaningful and gets out into the community to make a difference. You present your findings at various forums and conferences. You're also expected to contribute back to the field, either as committee membership or grant review, peer review, various things you can do for your community. Mentor young clinicians or scientists, and amongst that you need to manage your research team who do all the work. So as for me, this is me in high school. I always loved science. from when I was quite young. I watched my grandfather die of cancer when I was 12, so I always wanted to do cancer research. So this is me making a periodic table of the elements out of Tim Tams for my chemistry class. I then went to UQ and did my bachelor's degree and honours. Then came straight to QIMR when we were just one building, and was a research assistant for a year, and then ended up starting my PhD in the same lab. I went overseas for some postdoctoral training in Philadelphia, and then came back, and I've been affiliated with the hospital as what they call a senior scientist, so with Pathology Queensland, but based in QIMR ever since then. So as I said, you've got to plan to have children at some point if you want to do that. They're quite the undertaking. So I did that around this stage. I've seen a lot of clinicians think about when to have their family in amongst their training or a PhD if they want to do that. And there's a lot to think about. So I did that, and I now am a group leader at QIMR Berghofer, and I have my own team of people. You can see the institute is a lot larger now, so that's very exciting. And my children are quite a bit older, and they take care of themselves now. So during this time, I've been involved in grants that have funded over $30 million of research, and about a third of that has come directly to my lab. I've supervised a lot of students, honours and masters, and undergraduate students, 11 PhD students, and 4 of these are clinicians, and almost up to 100 publications. And what you'll come to see is that the citation of your publication is what's really important, because that means other people are reading your research and referring to it. So very early on, I came across Professor Barbara Leggett, the first lab I worked in after my degree. She was the lab head. She's an incredible gastroenterologist. She was the first female gastroenterologist in Queensland, and she's very clever and has run a research lab for many years in conjunction with, partnered with a scientist. So I did my PhD with Barbara, and I've put some pictures of other people here. Professor Laurie Powell, who is also, he's the pointer. Anyway, he's the older man. So he really was responsible for my first job at QIMR, which was working with Graham McDonald, who was the younger man, I was assisting him when he was completing his PhD. He's a hepatologist and previous director of gastroenterology at the PA. And I've put Professor Powell in here because he was always a massive advocate of clinician research and supporting researchers and clinicians. He was very inspiring. He spent most of his career working with a scientist, June Halliday, and they were a science clinician team and were incredibly productive. And so that was really my model for how I wanted to work. And the young man below is Dan Worthley. He was my first clinical PhD student. He's a gastroenterologist too. So he's gone on to have a private practice, but also is a very active researcher, and last year had a science paper. So this is Professor Jeremy Jass in the middle, and it was quite nice for me to come into this building because he was in Brisbane in the early 2000s when I was doing my PhD. He's an incredible mentor. He was the head of pathology at the time, and his office was here, and I used to sit opposite him at this microscope, and he really taught me pretty much everything I know about reading slides and looking at tissue. So he was very creative and taught me about seeing things under the microscope and then turning that into research projects. So I will show you shortly some of the work he was involved in. I'm going to skip ahead a bit because I'm really chatting. But here are some more people. Pauline Hall is the older lady here. She also was a very important mentor to me a very eminent pathologist who encouraged me to attend clinical meetings, which as a scientist is a real honour, and attend meetings where they're discussing patients and looking at patient samples. Andrew Cliston started Envoy Pathologist, which is a really internationally regarded, highly successful private pathology practice, GI pathology specific, and he has encouraged all his staff and pathologists to work with scientists actively, which is really important. And the bottom row there are my 3 pathology PhD students who have completed, and I'm still actively collaborating with all of them. So as I've moved on in my career, I've moved away from early colorectal cancer, so polyps, which are more a gastroenterology research problem, and more to how can we treat cancer patients better and more specifically. So about 10 years ago, I started working with Dr. Matthew Burge, the top man, Melissa Eastgate and David Wild, who are oncologists here at the Royal. And now I also work with David Clark and David Cavalucci, who are colorectal and hepatobiliary surgeons for collecting fresh samples. So almost get a bit emotional showing all these people because they've been so supportive. And if you want a career in clinical research, I just really encourage you all to seek out successful, generous people who usually I think the really successful researchers and clinicians are very generous and do want their work to continue in the next generation and do want to mentor young people. So don't be afraid to seek them out and speak to them and ask them what they're working on. So together we've published a lot and built a body of work that has contributed to changing clinical practice in a number of fields. So I'll start with the gastroenterology, which was in the early 2000s, and this was mainly work with Barbara Leggett and Jeremy Jass. So on the left-hand side is what we call a conventional adenoma. You can see it's quite an obvious bulbous polyp with a fairly typical adenomatous morphology under the microscope. And in the early 2000s, these were the main polyps that were considered a problem. Sorry, this is a colonoscopy picture in a bowel. So these polyps were always removed. They take about 10 years to become a cancer. And then mainly through the observations from Jeremy, he saw that there were other types of polyps. You can see here, you could almost not see it. It's a serrated polyp, called so because of the curly architecture. So he noticed these little polyps, which people would find and not even always remove at colonoscopy because they were thought to be inconsequential. And with the instrumentation at the time, you often couldn't even see them anyway, so many weren't removed. But together we made collections of these polyps and really made a difference to surveillance and screening and outcomes in this field. So we realised that these curly or serrated polyps actually precede around 1/5 of all bowel cancers. So, and they grow really fast. The adenomatous polyps take about 10 years to become a cancer, and the serrated polyps, depending on the age of the individual and other things happening, they could even become a cancer within 12 months. So you often would see what you call interval cancers. So in between colonoscopies, a patient could still get a cancer, and often it was one of these serrated types. So colonoscopy instrumentation has dramatically improved as a result of this work from us and other teams around the world. There's new guidelines for how to manage these polyps. We've worked with these exceptional pathologists who have refined the diagnostic criteria for the polyps. And our work has been cited in clinical practice guidelines and textbooks. And this is really gratifying to see how something you've worked on can actually influence clinical practice. This is more basic science work. So we did some more work in the lab, animal studies, and working with patient samples to try to work out the natural history of these lesions. This is an example of some of the specimens that we work on. So this is Jennifer Brodsky, my PhD student, who is now actually head of pathology at the PA hospital. So she helped collect all these samples where we identified the cancer region of the tissue and made what we call tissue microarrays, so we can look at a large number of samples in one experiment. And we can use these samples to look at all sorts of morphological features. This study was to look at immune cells. We can also look at the genes that are expressed on an individual cellular level. In oncology, I was involved in a study in 2008. A manuscript was published that was incredibly exciting. There weren't many treatment options for patients with bowel cancer, and there was a new drug called cetuximab, and they worked out from this study, which was led by a man in Adelaide, Chris Karapidis, an oncologist, and he showed that if you have a mutation in a gene called KRAS, you tend not to respond to this new drug, whereas if you're wild type for this gene, you have a much better chance of responding. And this was the very first predictive biomarker for bowel cancer treatment, so it was hugely exciting. And by the very next year, Merck had formed an advisory panel, and I was a member of that, and my lab actually generated and delivered the first 100 test results for KRAS in Australia. And there's really been very little advances in terms of oncology treatment and predictive biomarkers since this time. So that's what I'm working on now, how to better predict response to therapies for patients with cancer. So you customise the therapy for the patient so they're more likely to respond in the first instance. With metastatic bowel cancer, outcomes are really quite limited, so I think it's very important to give the best therapy for them up front, also to reduce toxicity. So we've been working on what we call organoids, which this is cell culture. We take a little bit of cancer sample at the time of surgery, and we can grow it in the laboratory. And this is a really accurate way to understand each patient's individual cancer. This is just to show you that it's a 3D structure that really recapitulates the biology and form of cancers in the patient. And these have been grown to test drugs. There's clinical trials that are ongoing to test drugs using these samples to predict response to available therapies. We've also started using these models to co-culture with immune cells to try to predict response to immunotherapies. So this has taken a lot of work to get up and running, and a lot of close work with our clinical collaborators. So David Cavalucci in the top is the hepatobiliary surgeon. So he collects liver samples for us at the time of surgery. So these are colorectal cancers that have metastasized to the liver, which is the most common site for colorectal cancer metastasis. Matt Bird, as I said, is an oncologist, and David Clark is the colorectal surgeon and collects colorectal cancer samples for us. So we collect these, grow the organoids, and freeze them down. We can do various tests and develop a lot of different experiments. So they're very useful. We call it a living organoid biobank. It's very labour intense to collect though. This is just to show the kind of results we get. TAS-102 and regorafenib are the last line therapies for patients with metastatic colorectal cancer. And you can see there's a blue and a purple sample, and the purple sample doesn't really respond to either therapy, but the blue patient was more sensitive to regorafenib. So this kind of result would tell the doctor to start with regorafenib for that patient. We recently, through all our work with our clinical collaborators and developing this organoid technology, we've been able to attract some funding for equipment and have started up an organoid facility for high throughput drug testing. And, this just shows you an example of a robot helping us out doing the work, and can actually test 384 samples at once with a lot of different drugs with very, very precise measurements, much more precise than we could do ourselves. So this robot is able to set up the experiment, put it in an incubator for the cells to grow. When it's finished, take it out by itself, put it in an imager, image the results, and tell us which drug to use. So we are doing a couple of preliminary observational type trials. One we call the OPTIC trial, which is the liver sample, so it's the metastatic samples, and we're working out a pipeline which we hope to deliver to patients in the future for working out this kind of sensitivity to therapy. And one thing I really forgotten about that I really wanted to highlight is the role of the clinician in these kind of studies. Obviously, sample collection is crucial, and annotating the clinical information and understanding the question is crucial. But this is an example where I was very excited when we first developed this technology, and I thought, wow, I'll test 1,000 drugs and work out exactly cure every patient, and I was quite happy with myself. And the oncologist said to me, well, Vicki, that's all very well, but the patient won't get that drug. If you have some obscure drug that's not FDA approved, they can't have it. Or if it's not TGA approved, maybe they can have it, but they won't be able to afford it. Or, then there's the issue of financial toxicity. So that's really influenced my studies, and currently we're just testing the available standard of care therapies because that's probably what's most useful at the moment. We have a rectal cancer project, which has quite a different biology and treatment strategy, so we're looking at those samples separately. And Kiki Lianos is actually in the audience today, so she's leading this project for her PhD. And finally, I think this is the final thing I wanted to show you. This is an example of how a clinician massively influenced my work, and this is not a unique example. This has happened time and again over my career. So Matt Burge came to me one day and said he knew about a different cancer type where they used a different drug, but he had related it to a similar drug available for bowel cancer, and he had the idea that maybe it would be good to try it in bowel cancer. And the drug was regorafenib, which is TGA approved but not on the PBS, which, as I said, is the last line therapy. It gives about 2 months survival in the refractory metastatic setting, and is very expensive and associated with a lot of toxicity. And he thought that combining it with aspirin might be effective. And just quickly, these the graphs where you see the purple line where the growth is much lower, it had an extraordinary synergistic effect of regorafenib and aspirin killing the cancer cells. And this is another example where we then took it to mice. So we did in vivo work, and you can see the purple line, which is a combination therapy, is pretty much a flat line, which means it pretty much killed the cancers. It was an incredible result. And the morphology here is just to show the healthy tumour in the mouse compared to the one on the right, where there's very few malignant cells left and most of the tissue is necrotic. So we thought this was very exciting, and we then, through Matt's connections and now my connections, I chair a translational research committee with the Australasian Gastrointestinal Trials Group, and Matt's also very involved in this committee. And they've helped us develop a clinical trial to trial this combination. And they've put us in touch with Bayer, who's agreed to fund the regorafenib, and helped us develop a really slick clinical trial protocol with statisticians and community members and so on. So I'm very excited about that and hoping it will go ahead soon. So the last couple of slides. So for you today, you need to think about what are you interested in. I think this kind of career is most gratifying when it's an area you're very excited about, and then finding out what's the biggest problem in the area, and that's an incredible contribution of the clinician to identify the problems that the scientists often aren't really savvy at doing. How can this be addressed, and is there someone you can work with? To build your research program, you need not just ideas, but a lot of money and staff and experiments, publications, sample repositories often, which can be very labour intense to collect. So this is my final slide about what can a clinician contribute. And I think my career is anything to go by. Clinicians are essential to the clinical research process in identifying the question and understanding the areas of need, accessing the patients and the samples, and then also, very importantly, using their contacts and knowledge and networks to drive translation and implementation of the findings. And I think scientists are also very important because they have knowledge of the scientific method and their established network for doing experiments with equipment and institutional structures, grant writing experience and knowledge of funding schemes, and time. So I think the greatest challenge is time. Clinicians, if you want to be an actively working clinician, you won't have a lot of time for research. As a full-time scientist, I really struggle to juggle everything I want to do and meet my deadlines, and so I don't know. I've met extraordinary clinician researchers who do it on their own, but I don't know how. So my final question is, can you do it all? I think you can, but I think you can achieve more together, and I'll leave it at that hopefully we can work together to achieve more translational research.
Thanks, Vicki. That's a very powerful story and a real testament to the perseverance, but also your ability to bring networks together. I think we've heard the message really clearly that for clinician science really to succeed, it is a collaboration. You need clinical insights, you need patients, and you need scientists to bring it all together. We do have time for one quick question. That's okay, because you actually probably answered a lot of questions as you went. Yes.
Thanks for that. That was an excellent talk. Really exciting about the organoid research aspects there. It seems like you've developed essentially an incredibly high throughput way of optimizing treatment. And you have the potential to develop a massive data bank associated with this. Do you have any genetic or epigenetic data associated with each individual patient?
Yes. Not the most recently collected samples, but we do. That's a great question, thank you. So that's really one of the biggest benefits of having banked tissue and biobanks. So we have many thousands of samples in the freezer that we've worked on for many years, where we have often whole genome or whole transcriptome, so we know about every gene in that sample, and we link that to clinical outcomes and detailed morphology from the pathologist. So that's very powerful and very time-consuming to put that together. So that kind of resource has taken decades to establish, and now, as you say, we're adding to it with the living organoids.
Yeah, no, excellent.
So,
Yeah.
sorry, so that's the kind of thing a clinician could come in as a small project to evaluate those sort of things that have been collected and make a meaningful project by building on something established.
So Vicki's taking masters and PhD students right now, so come and talk to Vicki. In the interest of time, Vicki, you'll be here at morning tea, will you?
Yes, I'm staying for the day.
Excellent. So you'll have the periodic table with Tim Tams ready for us out there. Fantastic.